dim light source not enough durable and long lasting. Okay, so you have N for nucleotidase, aminopeptidase, dextrinase, isomaltase, maltase, lactase, sucrase, and nucleosidase, enterokinase, dipeptidase, lipase. Clear? Clear? Yes. Next is process of digestion. Digestion is mechanical and chemical process. It's both mechanical and chemical process. Right now. Carbohydrate digestion. Write down carbohydrate digestion. In the carbohydrate, you know the carbohydrate digestion begins where? Where is the carbohydrate digestion begins? Are you all there? Mouth. Very good. So which enzyme is involved? Which enzyme is involved in carbohydrate digestion? Which enzymes is involved in carbohydrate digestion? Come on, tell me. Salivary amylase. Which ion is required for the activation of salivary amylase? Which ion is required for the activation of salivary amylase? Who will answer this? Chloride ion. Very good. So, very smart. So, chloride ions. So, write down where digestion of carbohydrate takes place. One is in the buccal cavity and other is the small intestine. There is no digestion occurring in the stomach. Carbohydrate digestion in the stomach. Very important question. Please write down this chart. Next, after write down about the carbohydrate. Done. Okay. Next. Next is protein digestion. Is there any protein digestion in the mouth? So protein digestion starts in stomach. Is it renin present in the adults? Very good. Write down this. Because casein is present in the form of milk that needs to get digested, it's a protein. So it's a complex food that needs to get digested to get into simpler compound. That's into amino acids. It's okay, no problem. No sorry. You have doubts, you have right to ask me. Done? Okay, next. Protein. Protein in stomach get degraded into proteoses and peptones. 50% of protein. Okay. Next, by enzyme pepsin, pH 1.5. Next, 50% protein digested in duodenum by enzyme trypsin and chymotrypsin to give what? Peptones and proteases. Here also it is in stomach. pH 1.5. Trypsin and chymotrypsin. Again the proteose and peptones, they digest in duodenum in presence of pancreatic, that is aminopeptidase and carboxypeptidase. Aminopeptidase and carboxypeptidase to form dipeptide plus free amino acid. Next, dipeptidase form free amino acid by dipeptidase. This is how finally you get amino acids. Done? Okay, next. Fat digestion. There is no stomach. Gastric lipase hydrolyze small amount of fat. In small intestine, fat is converted to fat droplets by the help of emulsification, and pancreatic lipase will act to form diglyceride, diglyceride to monoglyceride, monoglyceride to ultimately fatty acid and glycerol. Write down this about the fat digestion. What about for others? Are you able to see the screen? Komal, re-log in and come. Re-log in and come. The new students who have joined recently, are you understanding the concepts? We are talking about the digestion of food. The digestion of food is both mechanical and chemical. Mechanical is by the movement of teeth and also with the help of the peristalsis movement. Whereas chemical includes the different enzymes that are present in the different part of the intestine. Is it clear? We have studied the enzymes that are being secreted from the diglyceride too. I didn't get your question. What? Diglycerides to monoglycerides. Monoglycerides then again break down into fatty acid and glycerols. Enzymes is same lipase enzymes. So as it is lipase enzymes. Yes, intestinal lipase. No, no fat digestion in the mouth. Only and in the NCRT book there is a chart given. Okay, I want you all to refer to the chart with respect to digestion and enzymes. Is it clear? Are you all done? Can we move to the next? Then you have nucleic acid digestion. Nucleic acid digestion happens only in small intestine. Okay, not in mouth, not in stomach, not in other part of the intestine. It's only in the small intestine where nucleic acid converts into nucleotides, nucleotides to nucleoside, nucleoside to sugar and base. Enzymes, nucleotidase, nucleosidase are the enzymes that leads to simpler compound that is sugar and the base. Enzymes I had mentioned nucleotidase, nucleosidase. Clear? Is it done? Hello. Okay, next. End products of the digestion is carbohydrates is glucose, fructose, galactose, proteins, amino acid, fat, fatty acid, and glycerol, and nucleic acid is sugar and base. Nucleic acid is sugar and base. Write down. Done? Any doubts until here? See, the complete digestion of food takes place in the duodenum. The fully digested food is a semi-fluid in nature and it's known as chyle. The food that is present in the intestine, small intestine, is called as what? Chyle. Complete digestion of food takes place in the duodenum. The fully digested food is semi-fluid in nature and it is known as chyle. Okay. What is your question, Patricia? Aminopeptidase is used for nucleic acid breakdown to form nucleotides. No, aminopeptidase is for protein only, dipeptides. And what is chyme? The stomach stores the food for 4 to 5 hours. The food mixes thoroughly with acidic gastric juice to form the paste, and this is called chyme. Can you see the slide of a chyme?
No, ma'am, we can't say anything. We can just see that. And now we can see.
Chyme. Can you see it now? Which slide are you able to can see now? Okay, fine. Write down about the chyme. Are you done? Any difficulties there? So what is bolus? The teeth and the tongue with the help of saliva masticate and mix up the food into bolus. You should know what is bolus, you should know what is chyme, you should know what is chyle. Can you see digestion bolus slide? Can you all see the bolus slide? Hello. Are you all there? Not juice. Bolus, bolus. Okay. The teeth and the tongue with the help of saliva masticated and mix up the food into bolus. So you know what is bolus, you know what is chyle, you know what is chyme. Deglutition is swallowing. When you take in the food and you swallow the food, that is called deglutition. It's another term. Any other doubts? All clear? Can you see the digestive juices? Saliva, one juice, one enzyme. Gastric juice, three enzymes. Bile, no enzymes. Pancreas, six enzymes. Intestinal juice, 10 enzymes. Very, very important. Okay. And next, Okay, the next is absorption of food. We have discussed about the digestion of food. The next is absorption of food. Absorption of food. Write down absorption of food. Absorption of digested food. Absorption of digested food. Absorption of digested food. Okay. The first one is carbohydrate absorption. Now, what are the simpler units of carbohydrate? Glucose, galactose, right? Yes. So, glucose and galactose get absorbed by active transport. Active transport. Fructose by facilitate transport, facilitate diffusion. That is passive. It is passive. Next, protein absorption by, that is amino acid absorption. Maximum amino acids by active transport, some amino acid by facilitate diffusion. This is passive. It's passive. Now, absorption of fat. This is important. We need to understand here. Absorption of fat. That is monoglyceride, fatty acids, and glycerol. These are the simpler units, right? Fats are hydrophobic. Fats are hydrophobic. Hydrophobic means water love or water hatred? Water hate. Fats are hydrophobic. So fat, so they need bile salts, right? So they need bile salts. Where is it? So they need bile salts. Fat plus bile salts help in formation of micelle. So example, this is micelle. Too many molecules of fat is held together, forming a micelle. So this is micelle. This micelle we'll write down and I'll explain you. Okay. Enter bile salt separate. So then reformed in enterocyte to form triacyl glycerol. The next step is chylomicron formation. Chylomicron formation. What does it say? Enter enterocyte by simple diffusion. Very easy to understand. Write down. First step is this. Second step, then third step, then fourth step. Very easy. First you write down and I'll explain you. Done. Next. Let us draw this, it will be more clear. Can I explain? Are you done writing? Are you all writing it down? Can you see it clearly? Is it done? Are you all there? Can you hear me? Okay. Are you done writing? Do let me know. Once you're done with the writing, do let me know. Done? Okay, fine. Now listen to this. See, fat is a water hydrate. So fat cannot dissolve in water. So it needs a bile that emulsify and form these small globules. This measles now enter into enterocytes. Enterocytes are the cells lining the small intestine. By simple diffusion with a brush border epithelium, there is enterocytes into which this meseal granules enters. After it enters in the diagram, you can see bile salts get separate. Then other side you're getting triacylglycerol. This triacylglycerol enter into ER and Golgi apparatus of the enterocytes and then form a chylomicron. Chylomicron is a protein covering outside and inside there is a fat. Now it's a protein. Protein can be easily passes through. So the outer side is a protein covering. The inner is triglycerol or fat fats or phospholipid. Chylomicron are the lipoprotein particles, and this is formed by triglycerol, cholesterol, phospholipids, fat soluble vitamins covered by protein layers. This chylomicron easily get absorbed into lacteal cells. Do you remember the lacteal cells? If this is the lacteal cell, if this is the blood microvilli, and if this is one of the enterocyte, okay, like this, there are many enterocyte cells, and here is the blood vessel. This is blood vessel in the microvilli, and again here is what, lacteal in the microvilli. So this chylomicron, which is formed inside the enterocytes, get absorbed by blood vessels of the lacteal cells, lacteal vessels. This chylomicron, which is formed inside, will get absorbed by blood vessels or the lacteal vessels. Come on, tell me. Did you understood the question? The chylomicron that is formed in the enterocytes get absorbed by blood vessels or the lacteal vessels. By the lacteal vessels. Is it point clear to all? Any difficulties there? Okay. I'll give you some time. Go through it, entire thing what we have done for today. Everybody please go through it. Everybody please go through it. Yeah, Deepika. Protein absorption. Can you see? Everybody please go through this entire thing what we have studied today. The new students please go through and let me know if you have any difficulties. See, active transport is where there is a use of energy, and passive is where there is no use of ATP. Write down under the note all monosaccharides and amino acid are absorbed into blood capillaries. Write down all monosaccharides and amino acids are absorbed into the blood capillaries, whereas fat is absorbed into lacteals. Fat
Can you
is
repeat
absorbed.
once again?
Yeah, all monosaccharides and amino acids absorb into blood capillaries. Fat is absorbed into lacteals. Fat is absorbed into lacteals. Water absorption is by osmosis. That is passive. Water absorption is by osmosis, that is passive. Next, iron and calcium is absorbed maximum in duodenum. Iron and calcium is absorbed maximum in duodenum. Iron and calcium is maximum absorbed in duodenum. Are you all done writing? Jejunum is a site of maximum absorption. Jejunum is a site of maximum absorption. Jejunum is a site of maximum absorption. Ileum, vitamin B12 and bile salts absorbed in ileum. Vitamin B12 and bile salts absorbed in ileum. Done? Yes. The next. Hormonal control of GI tract. Write down. Hormonal control of GI tract. First is name. Second is source. Third is target organ. Fourth is function. The first hormone is gastrin hormone. Gastrin. Source is gastric mucosa. G cells. Target organ is stomach. Increase gastric secretion. Next. Gastric inhibitory peptide. Gastric inhibitory peptide or GIP or enterogasterone. Source is duodenum. Target is stomach. Decrease gastric secretions. Third is duocrinin. Duodenum stimulate Brunner's gland. Enterocrinin. Small intestine. Stimulates crypts of tubercle. CCKPZ or cholecystokinin pancreozymin. Small intestine, gallbladder, pancreas. Function: contraction of gallbladder, increase enzyme secretion of pancreatic juice, increase We have missed one secretin. We have missed, right? I'm writing it down. Secretin, duodenum, pancreas, liver, and stomach. Increase pancreatic juice. Increase bile secretion. Decrease gastric mobility. Fifth one, this is a sixth one. Any doubts? Yes, scripts of liver code. COL is scripts of liver code. Is it clear? Any doubts? Any doubts? So good girls are decent, elegant with strong characters. You can give anything. Boys, no need to be. It's nothing good girls or bad girls. It's just for uh, understanding. Is it clear? Just to make it easy, we can give any mnemonics. Okay. There's no good girl, bad girl as such. So you can give anything, whichever the mnemonics you would like to. But yes, this is how that you need to remember the different hormone. Is it clear? I know that you don't, but still, let me clear from my side. Yeah, quite there, Pratyusha. No using shut up and all there. Okay. Any doubts? What about the new students who have joined recently for the class? Any doubts? Mm-hmm. Tell me. Are you all understanding well? Ma'am, are the notes on cockroach posted? Okay, about the notes on the cockroach, I'll tell them to post today. Okay? So, can we wind up the class for today? See you all in the next class. Until then, keep learning. There's a small part which is left, or the disorder of digestive system. We'll cover this chapter and then we'll move to the next chapter. Okay? Done? So, take care, everyone. Bye bye. You're welcome. Bye. Take care.